Hello, and welcome to a video introduction to Next Generation Metabolomics, brought to you by Metabolome Technologies, or HMT. Today's presentation will be divided into 5 main parts, starting with an introduction to HMT as a company. Beginning life as a small spin-off venture from the esteemed Keio University in 2003, Metabolome Technologies has evolved into the cutting-edge metabolomics analysis service company that we know today. While HMT headquarters remain located in Tokyo, Japan, we've expanded our business across the globe over the last 15 years, with sales offices in the US and Europe, as well as distribution partnerships across Asia. In order to understand our strength as a metabolomics service company, we must first understand what metabolomics is. As with DNA and genomics, RNA and transcriptomics, and proteins and proteomics, metabolomics is a snapshot of small molecules, or metabolites, within a given system at the point of assay. This metabolome can be thought of as a functional consequence of all the other combined omes, and, as a result, is a close indicator of a given phenotype, be it healthy or a disease state. Metabolites are small molecules that are involved in energy utilization within a given system. As the structures of several metabolites shown here begin to demonstrate, there is a wide variation in terms of metabolite size, chemical composition, charge, and function. Being able to accurately measure metabolites across different regions of chemical space, while maintaining a high degree of sensitivity, is necessary to gain a more complete picture of the metabolome and, thus, understand how given conditions affect a system. HMT has achieved this through our patented CEMS technology. CEMS, or Capillary Electrophoresis mass spectrometry, is a unique separation technique where a small volume of sample is injected into a thin glass capillary. When a charge is applied, metabolites will migrate through the capillary according to mass, charge, and ionic radius, or shape, of the molecule. This facet of separation sets HMT apart from LC-based metabolomic service companies. Even with specific columns, liquid chromatography techniques have a difficult time separating structural isomers like those shown above. In the case of glucose 1-phosphate, glucose 6-phosphate, and fructose 6-phosphate, each of these structural isomers has a distinct functional role in cellular metabolism and, having all three elute as a single peak, loses valuable information about the state of the system being assayed. Because CEMS separates metabolites by size, charge, and steric valence, each of these structural isomers is detected as a single peak, allowing discrete measurements for each. Along with high resolution, CEMS affords us a high level of sensitivity. Looking at the traces of several phosphorylated nucleotides, we can see some degree of noise as well as some shouldering effects in the LC runs, whereas the CEMS peaks are singular, clean, and have much higher intensities. Another strength of CE is its accuracy. While most LC platforms have RSDs in the 10 to 20% range, we can see here that many important metabolites covered in CE have minimal RSDs, allowing for more accurate biological interpretations. As alluded to earlier, metabolites cover a wide range of chemical space. No single methodology covers them in totality. To this end, we have developed LC platforms to complement our patented CEMS technology. By utilizing both of these separation methods, HMT has a higher coverage of metabolic space, giving a more complete picture of the metabolome and its applications. Over the nearly 20 years since HMT's founding, we have worked with clients across a broad spectrum of research areas, from food science to cancer research and clinical cohort studies. Our experts have a vast breadth of knowledge across different areas of science, and we are as much collaborators as we are a CRO. By utilizing our distinct platforms, which will be covered more later in this video, we are able to assess the identity and levels of metabolites that link closely with various diseases, as well as processes such as biomarker discovery, patient monitoring, and disease progression. A particular area of research that has gained a lot of interest in the past decade is the microbiome. The average adult human contains about 30 trillion of our own cells, and about 40 trillion cells which compose our microbiome. With this in mind, the importance of the health, composition, and signaling of this microbial reservoir cannot be overstated. To that point, the microbiome has been implicated in many facets of human health and disease, from IBS to Alzheimer's disease. As diverse as the composition of our microbiomes may be, so too are their functional metabolites. At HMT, we have developed platforms that cover as many of these important metabolites as possible. While a large portion of the microbiome is located in the gut, there are other niches such as the skin. Moreover, assessing different sample types such as biofluids, organ biopsies, and plasma is crucial in determining the role of the microbiome during disease treatment, for example. HMT's ability to assess different metabolite classes across a variety of sample types allows us to gain a deeper understanding of the microbiome and its role in human health. As I mentioned earlier, we at HMT have developed many platforms, each with considerations for the specific needs of researchers who will use them. From untargeted offerings to isotope tracing, there is something for everyone to be excited about. Some of our most used services include BasicScan. BasicScan is our flagship untargeted platform, utilizing capillary electrophoresis time-of-flight mass spectrometry to assess over 1,000 polar metabolites, with no restrictions on sample type. This is a great option if you don't know what you're looking for, and it often serves as an important diving board. Researchers will often find interesting and unexpected paradigms that will shape the future of their research. For an untargeted platform that covers a greater breadth of chemical space, we offer DualScan. DualScan adds a liquid chromatography time-of-flight mass spectrometry run to the normal CE run, allowing for probing and detection of hydrophobic molecules, such as fatty acids, acylcarnitines, and bile acids, that would normally be difficult to detect by CE alone. Additionally, this platform allows for targeted coverage of lipid mediators of inflammation via our MScan package option. Focusing down now to our targeted platforms, C-Scope is one of our most popular offerings. It allows for absolute measurement of metabolites by running standard curves for all 116 metabolites in the assay, which allows for the calculation of various metabolic parameters, like adenylate energy charge. C-Scope covers central energy metabolism, including the TCA cycle, penose phosphate pathway, as well as amino acids, an extremely valuable targeted assay for cancer researchers in particular. F-Scope is our isotope tracing service, which can be critical in understanding a particular pathway or disease state. Utilizing the sensitivity of CE, we are able to easily see mass differences of a single neutron above the normal M/Z peak for a given metabolite. By assessing and evaluating how the labeled isotopes stack up over time, we can gain insights into the metabolome in a way few other services can. F-Scope covers over 50 isotopomers, and any isotopic label can be used. One of our newest and most exciting platforms on offer is OmegaScan. OmegaScan is an extraordinarily sensitive platform, utilizing CE linked to a Fourier transform mass spectrometer. OmegaScan is a prime candidate platform for biomarker screening when linked with our deep dive protocol, which assesses all known and unknown peaks within a run. A word about our process. After shipping samples to us, we will analyze them in our labs in Tsuruoka, Japan. After 4 to 8 weeks, a report will be delivered to you which contains your data, along with statistical analyses, like PCA and HCA plots, along with student t-tests. Of particular note within these reports are our pathway maps. Pathway maps allow for the reintegration of your data into a biological context, allowing for easier interpretation as well as ready-made figures. Our experts will also set up a meeting with you to go over the data and work with you for whatever you may need. With many years of operation under our belts, we have had experience with many different sample types. If we have not encountered a sample type that you wish to submit, we will work with you to run feasibility tests, as well as develop best practices for metabolic analysis of your samples. Thank you very much for your attention, and please contact us if you have any questions. Have a wonderful day.